Hello, my name is Craig Poskus. This is Chapter 16, Host Defenses Part 2, continuing on from Host Defense Part 1 from last week. Going to move a little bit further into specific immunity and some characteristics of MHC and antigens and some examples like that. As for Biology 150, Mr. Dorkowski. So it's funny, whenever I first started doing these, I thought to myself, man, a 10-minute PowerPoint is going to take me forever. I'm going to have so much information on everything. I'm going to be able to cover so much in the chapter until I actually started to do it. And then I realized that it's not that long of a time and I can't really cover in great detail what I want to cover. So we'll just go ahead and jump straight into it. First thing I'm going into is specific immunity. You got two basic types that I've symbolized there with the arrows. You got acquired immunity, and then you got innate immunity. So acquired starts with the products of the B and T lymphocytes, also known as antibodies. They are very specific and react within 2 weeks. So they're typically pretty slow. The innate immunity, they're monocytes such as the macrophages, dendritic cells, and mast cells. They're non-specific and usually react within 4 hours, so they are much, much quicker than the acquired immunity. Let's move on to the next slide. It's going to be characteristics of immunity. There's 6 total characteristics. I've got 3 on this slide, 3 on the next one, just so that way the slides didn't get too wordy and there's too much to read. So the very first characteristic, if you can see it right there, I apparently cannot say this word. I've tried many different times, so we're going to let Siri do it.
Specificity.
So it is highly specific to the antigen against which the third line of defense is directed. Next is memory. It is the rapid mobilization of the lymphocytes that have been programmed to recall their first engagement with the invader and rush to the attack against. And then the last one is inducibility, and it's only turned on when triggered. And moving on to the final 3 characteristics, you got diversity, always at least one cell that can react with any type of antigen. The next one would be tolerance, does not react to self-antigens. And clonality, generates millions of cells within the same specificity. See, I told you I couldn't say that specific word. But moving on to the next slide that I have here, it's going to be MHC, which is proteins that are produced to help fight antigens. So they provide a major role in immune response. The T cell recognizes the antigen as a complex with the MHC. This requires the antigen to be processed, and then after that, the antigen will unfold and be digested before it complexes with the MHC. So there are 3 main classes of MHC proteins, and they vary pretty heavily between the 3 once you actually start breaking down the characteristics of them. So class 1, it starts with that each human will inherit a particular combination of MHC genes. It also has proteins that tell cytotoxic T cells to kill infected cells before it can infect others. It also allows for recognition of self and regulation of immune reactions. And then finally, this is one of the main differences between class 1 and the other 2, is that the markers will appear on all nucleated cells. And obviously next is going to be class 2 MHC. I just put the numeric 2. You could put class II. It doesn't matter as long as it's class 2. So starting with the code for immune regulatory markers found on macrophages, dendritic cells, B cells. In class 1, if you remember, it's only the nucleated cells. This one is macrophages, dendritic cells, B cells. Those are monocytes. The second characteristic is going to be helper T cells. What they do is they start immune response against the antigens that enter the body. And then, just to go a little bit deeper into it, the 4 main regions, including, the class 2 has 4 main regions that include a specific region that is highly conserved A2 and B2, and that binds to CED4. My apologies, I believe that's a typo. I believe it's just CD4. It binds to CD4. And last but not least, we have the class 3 MHC proteins. This is the third and final class. It includes several complement components, tumor necrosis factors. It's not a membrane protein and have no role in antigen presentation, although they do play a significant role in immune response. Also, there is several functions and structures that are different, referring to the proteins that are encoded in the third region of MHC. Okay, and this brings me to the next PowerPoint. What the whole subject of host defense is dedicated to destroying. This is the enemy of everything that I've talked about in not only this PowerPoint, but part 1 of host defenses. We're going to move into antigens, get a little bit deeper on it. So it is the foreign substance that enters the body. This, when it enters, will stimulate the immune system to form antibodies that react specifically to the type of antigen in the environment. So depending on what type of antigen comes into the body will be the ones that reacts to all the different types of host defenses that I've went over. The body will form active lymphocytes that will destroy the antigens, like I just stated. But not only the different types of defenses that the body has, all of them are designed to go after antigens. And finally, just a little chemical traits of antigens. Just pretty simple, just quick to go over it. It's proteins, polypeptides, lipoproteins, nucleoproteins, glycoproteins, polysaccharides, lipopolysaccharides. I think it's pretty impressive that I can say lipopolysaccharides, but I can't say the specificity word. And after all of that, that's going to push me probably over my 10-minute mark, and I'm going to have to go in and shorten some of the slides and speeches to get it to fit the time frame. But this is the final part of the host defenses. If you have any questions, comments, or concerns, please let me know. You have my email, or we can communicate through Canvas. For anybody watching, because I made this a public YouTube channel, I hope this helps anything. This is a microbiology course in a community college, but I hope it's informative enough for your classes. Thank you.